Hey folks, let's go over the chapter 6 PowerPoint together. So here we go. We're human biology, and we're going to talk about blood. We're going to talk about what blood does, what the formed elements in blood are. We're mostly talking about cells and platelets, which are fragments of cells. And we'll talk about what blood plasma is made out of. Anyway, let's jump into it. So the heart pumps about 75 milliliters of blood with each contraction. On average, if your pulse is where it should be, it's 70 times a minute. So your entire supply of blood is circulated through the whole body every minute. That's pretty good perfusion. Or I should say, pretty good circulation. It's a good rate of exchange. Everything goes through the body. It means everything goes through the lungs. It's nice. All right, so functions of blood. It moves things, it moves oxygen and nutrients to the cells, it takes waste and carbon dioxide away from the cells. It carries hormones around, which are chemicals. We'll talk about the endocrine system later, but they're chemicals that are used as messengers or signals between different parts of the body talking to each other. So that's the transportation function. For defense, it avoids having, it carries white blood cells around and antibodies around to look for pathogens, look for things that don't belong, and can help you fight them. So, and obviously blood clotting. We have things like thrombin that we have things like thrombin, things like fibrin that will help us with making blood clots that will stop us from having the blood all run out after we have an injury. Let's move on. All right, so regulatory functions. Um, Obviously, blood is super important for body temperature. If you're too hot, you're going to send that blood up to the surface, up to the skin, and you're going to sweat to cool off the skin, cool off the blood, and bring the cooler blood back to the body core. It also regulates your water balance. Blood regulates your water balance by controlling, the example they have here, proteins dissolved in the plasma maintain osmotic pressure and regulate water balance. We'll talk later about hydrostatic and osmotic pressure, but when your heart pumps, there's some pressure at the capillary that pushes fluid out of the capillary, at the front of the capillary. That's called hydrostatic pressure. At the other end of the capillary, you are pulling fluid back in from the tissue, and that's called osmotic pressure. So hydrostatic pushes out of the capillary into the interstitial fluid, into the tissues, and osmotic pressure brings it back. We'll talk about that more. The key thing to remember is that water always wants to dilute things. So if you put all the fluid out and you keep a lot of stuff in the blood gets more and more concentrated, and eventually the fluid wants to come back. All right. And the blood's full of buffers. There are 4 different buffering systems. The big one that we're going to be talking about would be the bicarbonate buffering system. It says keeping it a constant 7.4. In reality, your blood pH varies a little bit, but that's right dead in the middle. It goes from 7.35 to 7.45 under normal conditions. If it goes above or below if it goes below 7.35, it's way too acidic, and you're going to have problems. It doesn't even want to carry oxygen then. I don't know what the side effects are if you go above 7.45, but I know that's the upper bound, upper limit for normal range. Anyway, let's go on. So blood is a connective tissue. You might remember that connective tissues have 3 characteristics. All connective tissues, whether we're talking about cartilage or bone, or in this case blood, they have a ground substance of some form, and that here in blood, it's blood plasma. They have fibers of some type made out of protein. Fibrinogen, which turns into fibrin when it's activated, would be an example of a blood protein that is a fiber. And then all connective tissues have specialized cells. And of course, the blood has a ton of them. We're about to talk about them now. Anyway, to recap, all connective tissues have specialized cells, protein fibers, and a ground substance. The protein fibers and the ground substance together are often referred to as the matrix. All right, let's move on. Blood is a connective tissue. It's got the formed elements, which are red blood cells, white blood cells, and platelets or thrombocytes, which are pieces of cells. And those things are suspended in blood plasma. The formed elements are made in the bone marrow. So red blood cells are also called erythrocytes. It means red cell. White blood cells are also called leukocytes. It means white cell. And thrombocytes, I'm not sure the meaning of thrombo. I should look it up. But I would imagine it means piece of a cell because that's what platelets are. All right. So the next slide is figure 6.1, origins of blood cells. You can see that we have stem cells in the bone marrow, and those stem cells, they're what we would call pluripotent stem cells. The PL should remind you of the word plural because they can make more than one type of daughter cell. They can make any of the formed elements that are needed. There are other types of white blood cells which are more powerful. We call those totipotent. They can make totally anything. Anyway, but let's move on. Oh, to be clear, there are no totipotent cells that I'm aware of in bone marrow. Bone marrow has pluripotent stem cells. Moving on. All right, so in 6.2, they show what happens if you take a tube or a capillary tube or a vial filled with blood and you spin it down in a centrifuge. What's going to happen is all the formed elements are going to settle out. So the red blood cells will be towards the bottom or towards the outside of the centrifuge. Then you'd have this layer right here. It's this thin white line. We call that the Buffy coat. That's the white blood cells and the thrombocytes, the platelets. You see how there are way more red blood cells than there are white blood cells when you look at this. And then all the stuff above it is the plasma, which is thick, sticky fluid, lots of albumin proteins. All right, moving on. So that blood plasma we just mentioned, it's 91% water, 9% salts and organic molecules. A lot of those organic molecules we're talking about are proteins, and the biggest one in that list of proteins we've already mentioned, it's albumin. All right, so you remember that solutions have a solvent, that's what we dissolve things in, and solutes, the things we're dissolving. Here, the book says solutes help maintain osmotic pressure of blood. Fancy way of saying it, but the short answer is a lot of the proteins that are dissolved in blood are too big to actually leave the capillary. And what that means is that the osmolarity, the concentration of the blood gets thicker, stronger, gets more concentrated as you go through a capillary. So at the far end of the capillary, you are going to have more concentrated blood plasma, and that will draw water back in from the interstitial fluid. All right, moving on. We also have salts. The bicarbonate is one of those things from sodium bicarbonate. It's a huge buffer. You remember a buffer means it keeps the pH of the blood in a specific region. We already said in the case of blood, buffers keep all different fluids in their certain solution. But this buffer in blood is one of the buffers that keeps things at 7.35 to 7.45. All right, other things that are dissolved in the plasma: nutrients, waste, hormones. All right, where it says plasma proteins are the most abundant organic molecule, think albumin. Albumin is so common that it gives a straw color to the plasma. All right, and again, albumin is a major osmotic pressure thing. The albumin molecules are too big to leave the capillary. So when fluid leaves the capillary, the albumin stays behind, and it gets more and more and more concentrated as we go through the capillary and lose fluid. Eventually, it's so concentrated that the fluid wants to flow back into the capillary to dilute the albumin solution. So, all right, there's the osmotic pressure again. Let's move on. So 3 major types of plasma proteins. The albumins are the most common one. They contribute to the osmotic pressure more than anything else, and they're transport molecules. Globulins are globe-shaped. They're round, okay? And they transport different substances. Some of them, like gamma globulins, those are those fight disease. Fibrinogen, I mentioned that one before. If you see ogen on the end of a name, that means it is the inactive form that can be converted to an active form. So fibrinogen is the stuff that if we need a blood clot will turn into fibrin. What I'm saying is you have these rounded proteins that move around in the blood flow really easy. They don't do anything. They're not causing any trouble. You get to a spot where there's a cut and there are chemicals saying it's a cut, and all of a sudden they go all transformers on you, spread out into this long girder, and the blood pressure, blood trying to flow out the wound, is going to wash these fibers up across the cut. And they're going to be like the branches on a tree that falls across the stream. Everything's going to start piling up behind them. The stream, you see all these leaves, all this trash that piles up behind the tree that's across the stream. Well, here you're going to have all of the fibers and the red blood cells and everything you need to make a clot. That's going to back up behind these fibers. All right, so let's move on. Oh, here's a check your knowledge, check your progress thing. It's the 6.1 one. I am going to pause for a few seconds. You can put this video on pause, go check your knowledge, make sure you know this stuff well. And when you come back, when you've checked yourself, go ahead and restart this video and we'll continue. So I'll be quiet now for a few seconds. Okay, let's move on. So we're going to talk about the red blood cells. It says, explain the role of hemoglobin in gas transport. Most of the oxygen that is carried by hemoglobin. Hemoglobin has 4 globulins, 4 rounded subunits stuck together, and each one in the middle has a heme, and each heme is capable of carrying an O2 molecule. So what that means is that a hemoglobin can carry up to 4 O2 molecules. Anyway, and it says, summarize the role of erythropoietin in the red blood cell production. EPO, that's the abbreviation for erythropoietin. That is a hormone from the kidneys that will tell the bone marrow, hey, those red blood cells that you're having the process of growing up, speed it up, make it go faster. We need red blood cells. The body is having trouble getting enough oxygen. Give me some red blood cells. All right, so red blood cells are biconcave in shape to increase surface area. Lots of words. What they mean is that take a cell and imagine a donut, but the hole is not all the way through. Instead, so you have a round cell, but it's rounded this way. You turn it the other way, it's kind of flat. But it doesn't have a hole all the way through. It does have the sides concave, dished in the middle, getting close. What you're doing is giving the maximum amount of surface area that makes it easy to exchange gases, easy to pick up oxygen in the lungs, easy to drop carbon dioxide in the lungs. Those are the things you want to do. Inside, there's hemoglobin, and that's about it. A red blood cell, as it grew up, as it became a red blood cell, it lost its nucleus, it lost its mitochondria, it doesn't have much else going on. Now, there are some good things and some bad things about that. It makes sense to get rid of your mitochondria. If your job is to carry oxygen, you don't want the thing using oxygen on the way. So going to the tissue, it would be horrible if the hemoglobin gets there and, sorry, if the red blood cell gets there and it says, oh, well, I've used up all the oxygen for my own needs. So to avoid that, red blood cells don't have the mitochondria that would use oxygen. Red blood cells are basically nothing but a sack of hemoglobin. But that means they wear out real easily too. You're always replacing large numbers of them. I think, we'll look it up. I think the lifespan of a red blood cell is like 20 days. Anyway, don't quote me on that. That was back of my brain calculation. Need to confirm it. Anyway, so red blood cells are red because they're full of hemoglobin. They are brighter red when that hemoglobin is loaded with oxygen. They are kind of a dull brick red when the hemoglobin doesn't have that much oxygen. So each globulin has in the middle of it a heme. Each heme can carry an O2. Heme can also carry CO2. Anyway, so when you're bound to oxygen, we call it oxyhemoglobin, and it is like bright cherry red. When the oxygen has left the hemoglobin and it goes and the oxygen is off being used by tissues, then the hemoglobin's kind of a dull brick red, and we call it deoxyhemoglobin. You can totally see the difference. All right, so here we have red blood cells. You can see the biconcave disc thing. You can see the dish-shaped protyle and disc shape in the side of the red blood cell here. As they're going through a capillary, the capillary is so narrow, they literally are going single file. There are some capillaries that are so tiny that the red blood cells actually have to turn and go sideways through that tiny capillary in single file. Anyway, let's move on. So here we got a hemoglobin. You can see it's got 2 of this globulin molecule, 2 of those. That means it has 2 alphas, 2 betas. I don't remember which of these is which. But anyway, we've got 4 globulins. Each one has a heme in the middle. This is red because it's full of iron. Okay, hemoglobin has heme groups. Heme groups have iron, and the iron is precious. It's essential. You don't want to lose too much of that. Anyway, but each iron can hold an O2, an oxygen. So how do we transport oxygen in the blood? How do we transport carbon dioxide in the blood? About 7% of it is in the plasma and 23% is on the hemoglobin, okay? The majority of it is part of this bicarbonate buffering system that I was talking about. Here, this formula, I love this formula. This is super important. If you have carbon dioxide plus water, the 2 together form carbonic acid. Now, carbonic acid is a weak acid, and that means it's a reversible reaction. A strong acid just gives off its hydrogen, it's done. A weak acid, we can usually go back and forth either way as needed. And that's the key to how this forms a buffer, okay? If you look at that, you'll see that we can go both ways. We could take hydrogens out of a solution with by combining them with bicarbonate and forming carbonic acid molecules. Or we could go the other way, and we could break down carbonic acid to release hydrogen ions to lower the pH. So basically what I'm saying is if the pH gets too high, we're going to break carbonic acid down and give off hydrogen ions that will lower the pH. If the pH gets too low, we are going to take the bicarbonate, take the hydrogen ion, hook those two ions together, and make carbonic acid. The net result is that we're removing hydrogens from the solution. We're going to make the pH go up because we've taken hydrogens out of the solution. One way that we can do that is with the respiratory system. If you have a blood pH that is too high, too basic, you are going to breathe a little more slowly, a little more shallowly, without even thinking about it, as your body tries to not get rid of as much carbon dioxide, build up more carbonic acid to neutralize the basicness that's happening. If you go the other way, if your blood gets too basic and needs to be more acidic, wow, totally want to edit that. Can't edit that. Okay, so if your blood is too acidic, then you will shift to the left, taking hydrogen out of the solution by making carbonic acid. You'll probably do this. One method would be by breathing harder and faster to get rid of carbon dioxide. That shifts the entire thing, both reactions, to the left. If, on the other hand, your blood is too basic, you are going to shift the entire thing, both reactions, to the right by keeping carbon dioxide in the body, by breathing a little shallower. Anyway, but it all depends on this bicarbonate here that you either take out or add as needed. That buffer determines the pH in the blood. It's one of the four systems that does. All right, so bone marrow is where we make blood cells, okay? As red blood cells are made, they grow up, they lose their nucleus, they lose most of the organelles. I already told you that includes mitochondria. If you don't have that stuff, you can't repair the cells. Okay, here's the time. It's 120 days, not 20 days. So it takes 120 days. That's about the total lifespan of a red blood cell. As it gets older and stiffer, it has trouble wiggling through all those little capillaries. You don't want it to get stuck there. So instead, in the spleen, which is lots of twisty little capillaries, it gets caught in the spleen, and then you have macrophages, the big eater of white blood cells, come along and digest it. Okay, but the shape of red blood cells allows them to squeeze through capillaries, and it gives a lot of surface area for the gas exchange. EPO, erythropoietin, that is the hormone produced by the kidneys when you don't have much oxygen. That causes the bone marrow, it says, to produce more red blood cells. It's probably more accurate to say causes it to make more red blood cells available right now. It speeds up the maturation. I don't know if it speeds up the total development, like creation, but it certainly speeds up the maturation. You dump more red blood cells on the spot. Blood doping is a messy word. Your book says, blood doping is increasing the number of red blood cells, sometimes by injecting EPO to increase the stamina of athletic performance. That is a great way to write it, because sometimes is the case. In older days, they used to do it old school, like in the 70s. They would take out blood months before the event, like you're donating blood. spin it down to get rid of the plasma, and then stick the red blood cells back into the blood. Super dangerous because the blood gets really, really thick. There are too many red blood cells. We call that polycythemia. And that's why you'd have perfectly healthy athletes healing over dead from heart attacks. It's because the blood got too thick with all the extra cells that were added for the heart to even pump right. Really messy. So that's old school. Then we had the EPO injection, but that's what the Olympic Committee started testing for. So then after that, they went to looking for EPO precursors, because once there were good tests for EPO, then the trainers are like, I'm not going to give my athlete that hormone because they'll get tested and we'll get in trouble. I'll give them the chemicals that the body turns into that hormone. And that's what Lance Armstrong was busted for, actually. Anyway, but yeah, blood doping is a method of trying to increase performance, increase stamina. And one method for a while was to inject erythropoietin. We use this same technique for legitimate reasons. If someone has anemia and they go to a hospital, they're probably going to put them on a diet that has plenty of iron, but they're probably also going to give them EPO to have them make red blood cells more quickly and have better stamina. All right, let's move on. So if you're low on oxygen, your kidneys are going, ah, and they release EPO with sound effects, I promise. Okay, not with sound effects, but they will release EPO, erythropoietin, and that will make the stem cells in the bone marrow make red blood cells, or make red blood cells grow up faster. And then you'll have more red blood cells, so you carry more oxygen. So the oxygen level is solid and it makes the kidney happy. That is what we call a negative feedback loop. Most of the feedback loops in the human body are negative. What that means is, here's a situation, let's do the opposite and cut down on the bad symptoms. So negative. All right. Disorders involving red blood cells. Jaundice. That's that yellow, you might have seen like a nasty yellow hue around a bruise, like you have the black and blue in the middle of the bruise, and then around it you have this big yellow. If it goes extreme enough, it's not just skin, you have the whites of the eyes that could turn yellow. That's the red blood cells out of the blood in the tissues. Anyway, anemia is when you don't have enough red blood cells, and or you could have enough red blood cells, but none of them have enough hemoglobin inside of them. So iron deficiency, that's one of the things that can cause anemia because you're not making enough hemoglobin. Pernicious anemia is when you don't have enough B12 and you can't make red blood cells. Folic acid anemia, that's another cause. You're not getting enough folic acid. You're not making your red blood cells. Hemolytic, lyse or lytic. Lyse means to break. So if all your red blood cells are breaking open, that's hemolytic anemia. And then we have sickle cell anemia listed here. It's a genetic disease. It's caused by a point mutation, a single point change in what amino acid you use in the hemoglobin. And what that does is, under low oxygen conditions, the hemoglobin changes shape slightly and it sticks to other hemoglobin. You form these long curved rods of hemoglobin, and because of that curve, the cell, as the rod is growing, the cell gets stretched over it into this big sickle shape. And those sickle shapes stick together real easy, and they get stuck in capillaries, and they can cause tissues to not get any oxygen because of that traffic jam, and it can kill tissues. Anyway, so check your progress. Okay, we're back. Let's move on. So we're going to talk about the white blood cells. And I'll tell you right now, what you want to know is the acronym Never Let Monkeys Eat Bananas. Never let monkeys eat bananas. That's neutrophils, eosinophils, neutrophils, lymphocytes, eosinophils, never neutrophil, let lymphocyte, monkeys, monocytes. Monocytes are baby macrophages. Those are the white blood cells that grow up to be the macrophages. So what I'm giving you with never let monkeys eat bananas is neutrophils, lymphocytes, monocytes, eosinophils, basophils. Those are the types of white blood cells that exist in order from most common to least common in a healthy person. And one of the tests they do in a hospital is called a differential white blood cell count. They actually are looking like, hey, this person has an elevated number of lymphocytes. And they can tell, not always, but a lot of times, they can tell what's probably going on in the body just based on which white blood cells your body thinks it needs to have the most of to fight the bad things that are happening. Anyway, and then we can talk about disorders with white blood cells, things like leukemia, lymphoma. Moving on. So white blood cells are leukocytes. They're fairly big. They have a nucleus, or it says large cell, and I said fairly big. I do want you to know that lymphocytes aren't that big. They're bigger than red blood cells, but they're not huge. Anyway, but some things like monocytes, the M in monocyte should remind you that that cell is massive. Some of these white blood cells are really big. Anyway, they're clearish. They're not white, they're clear. And if we don't stain them, we can't even see them easily. So when we're preparing a blood smear and staining it, we typically use something called Wright's blood stain. Anyway, moving on. So these white blood cells are made in the bone marrow, and they're produced depending on the level of colony stimulating factor. White blood cells fight infections, and they're an important part of the immune system. Some only live a little while, a couple days maybe. Some of them can live months, maybe years long. Moving on. All right, so here are types. And you can see that they've divided them into ones that have granules. We call those granulocytes. And ones that do not, ones that are clear. Now, I will tell you, the granules in neutrophils are really small. By the way, you know it's in.
Hello. Good, how are you?